Well, we are seeing more of a trend generally looking for long-acting formulations. We're looking to extend the duty cycle of a drug, reduce the burden on the patient, improve the user experience overall, and increase in adherence as well. You're less likely to miss a dose if it's once every 2 weeks, or if you do, the PK sort of peak is not quite as affected in a long-acting formulation. So generally that means that we're also having to try and tune the release rate of drugs. And there's a lot of formulation technologies that therefore are sort of more brought into the fold now, of trying to tune the formulations to that ideal release rates for individual drugs, to effectively go along that sort of line of ideal versus actual release rate, moving it more towards the centre. It's memory, right? So, you know, we struggle to remember to take our pills every morning. Certainly I do. Something that's once every 2 weeks that I can put into my calendar, that maybe it's something that I need to keep in the fridge, maybe it's something that I need to prepare beforehand, and that's certainly true of the colloidal dispersion. And we're going to talk today about some of the challenges associated with the colloidal dispersion. If I have to spend 5 minutes preparing it, that's quite a burden on my day. If that's every single day, if it's once every 2 weeks, that's going to be a lot more acceptable for a patient. So trying to reduce the regularity that a dose is required is something that's core to improving that patient experience, and probably core to improving the adherence as well.
So suspensions are challenging because they often like to not be suspended anymore, is a short way of putting it. If you leave some suspensions for a long time, they will settle and you will get an accumulation of drug on whatever is the bottom side of the device. And they tend to clog a great deal more. Whether they do either of those things is possible to predict in some cases, is harder to predict in some others. So there's quite a lot of regimes of different behaviors, but the sort of fundamental job that we have to do is to try and take one of these formulations and try to predict, first of all, whether it will either settle out or clog or do anything like that, and second of all, to mitigate that if it will in some way or other.
Yeah. So there's a few things about trying to get it out of the vial, really. Yeah. It's that getting agglomerates through a small needle.
Yeah.
It might be even settling on walls as well. You know, taking something that has settled onto a wall, and also creating a homogeneous solution as well. You know, a colloidal dispersion rather than a colloidal sort of settlement. And all those are quite differentiated challenges, I'd just say.
Sort of the first question you want to ask usually is something like, how big is the particle you're trying to put in? And if it's bigger than the needle, it'll probably block. Yeah. And if it is bigger than the needle and you cannot get away from that, then you might be able to get around it by having very non-spherical or very squishy particles. You might still be able to deliver them then. As you go smaller, they might still clog for purely mechanical reasons, but once you get below a certain size, they mostly clog for adhesion reasons. So they adhere to each other, or they, as you were saying, they adhere to the walls of the device or to the walls of the needle wall. And predicting that adhesion, there's some sort of there's some fairly well-established theory helping us to predict that. In some situations, it has limitations. So it's about applying those bits of theory, DLVO theory, and in some ways trying to expand it as well in certain situations. There's an increasing effort now to try and expand DLVO theory to non-polar, or in other words, non-aqueous solutions, so putting it into oil-based solvents and things like that as well. It turns out you can actually use that theory in a similar way. But the fundamental question there is still, does the stuff stick to itself or to the walls of the container? Because if it does, then you're probably going to be in trouble. Yeah,
and we continue to bring it back to the formulation. I mentioned earlier about tuning the release rate and the different kinds of particle technologies, or even overall dispersion technology, such as oil-based solutions or lipid particles, or you know, tuning between the nano range and the micro range in terms of your small spherical particles. In each of those cases, there's a different fundamental set of challenges there, isn't there?
Yes.
I mean, even in mixing technologies, going between an oil-based dispersion and an aqueous-based dispersion. There's a very differentiated challenge there in sort of shear transfer through the fluid, in overall viscosity challenges, and then we go down to how injectable is it, what kind of effect does it have on the tissue when you're breaking through as well. So it's all contextualized by those formulation technologies.
Yeah, it is. Viscosity is quite an interesting one. If you have very high viscosity, then it can help you to prevent things like settling out. It can mean that the settling time is, a year or something, which means that effectively it doesn't play a role. But obviously, if you do that, then it means that getting the drug through the needle is going to get harder. You need to push harder, and then if you increase the viscosity, it also tends to have a pretty strong influence on pain as well. So you get this sort of competing effect, which is normal enough for an engineering problem.
Exactly, it's that drug-device interaction that we see with every combination product. It's it might be easy that the formulation doesn't quite settle out of solution as much, but equally now we're looking about a much higher injection pressure, and now we're starting to think about polymer creep with a compressed spring for that period. So there's definitely trade-offs. The traditional method of resuspension is shaking, and we're all used to shaking up colloidal dispersions from a protein shake that is effectively an aqueous colloidal dispersion to some of the drugs that you might have prepared in every day, and traditionally that's come from a vial, where I've got a vial of a dispersion that I need to resuspend. There might not be anything in there. It's just the fluid, and the fluid's got enough room to move so that I can resuspend it. Now that's a very manual thing, and it's a very user-centric method of resuspending something, and therefore between people there's a lot of difference. The way that I shake might be fundamentally different to the way that you do, and that might be in the axis, that might be in the severity or the overall action. And we've seen from some of our experimental work in the past that people can shake up to 35G, whereas other people at the other end of the user scale might be as low as 5G. It's a large difference. So there's a few things you can do to change the way that mixing is done and in shaking, but fundamentally it's all user driven. Whether you're putting in a large ball to aid some of the deagglomeration or to improve local shear, it's fundamentally driven by the user. So we then look to this as more of an engineering challenge. Can we take that variability out of resuspension as much as possible, and that typically goes into building functionality into a device, whether it be there's a number of solutions on the market that look to transfer fluid through a restriction from one vial to the other, and you might have two different fluids. You might have powder in one and fluid in the other, and that moving it back and forth, you create sufficient turbulence that there is reasonable mixing. But again, we go back to the context of your formulation. The way that you might wish to mix with one formulation will be vastly different to another. The way that you wish to mix a biologic, for example, might be fundamentally very different, because biologics are incredibly sensitive. Certainly those encapsulated, for example, in a lipid nanoparticle. It's shear sensitive. And therefore, the way that you mix that, even if it's poorly wettable, needs to be thought through quite considerably different. And it's not just a challenge of variability in people. It's also messaging to people of what the differences are in your formulation. For example, there's 3 different animal drugs from Zoetis that all have very similar names. They all look the same, all colloidal dispersions. One will recover over 90% of homogeneity within 5 seconds when you shake it, another 10 seconds. And the third, which sounds immensely similar and has otherwise similar handling requirements, is over 90 seconds to get that recovery. So the variability comes in your messaging, the variability comes in what you're asking the user to do, and it's all contextualized on what that formulation needs to be.
Yeah, that kind of variability you were talking about can to some extent be predicted, in that you can say something about the amount of energy that it's going to take to break apart the attractive forces that have brought everything together in agglomeration. and compare that to the amount of energy that a human is ever going to be capable of putting in. So you can sort of immediately put yourself in the ballpark of, will they even be able to shake it loose, or will that just never work? You were talking there about sort of mixing and chambers, and that brings the question in of primary packs, which is a kind of whole extra area of risk that some people will have a desire to try and fix and deal with, and some people will not want to touch. So if you want to introduce complicated flow paths which mix and introduce turbulence perhaps, if you can, that usually involves changing the primary container, and the appetite for that varies quite a lot. And if you can't do that, then you're looking more at things like acoustic things or ultrasonic things than shaking. But you could also possibly put things in the primary pack. That's a kind of halfway house of a material that you are definitely going to have to validate and sort of show that it's entirely biocompatible and safe and all the rest of it, but it's not changing the primary container in quite the same manner as introducing a whole new structure. So
we're not necessarily changing the filling method in that particular case, which is another thing that's quite important. And we don't really want to change it unless we absolutely have to. And as I say, using primary packs again, inline mixing methods perhaps, depending on the fluid velocity, something that we could think about as well.
I think I would desire strongly for them not to be competing. They usually work best with each other. Any analytical model is going to be full of assumptions. It's going to be limited in terms of its applicability. And so you need usually some kind of experiment against which to validate. Not quite always, because sometimes it's happened already, but often. And equally, doing experiments can be quite time-consuming and quite expensive. And so if you have an experiment against which you validate a model, and then you use your model to sort of extend the sort of parameter space, I suppose, that you investigate slightly beyond, and so it reduces the number of experiments perhaps that you feel you need to do in order to be sure that you've found the solution you want to. They sit with each other. They're complementing each other. I would say making that judgment on an individual project is quite fun and quite tricky as to where exactly you want to sit. But yeah, that's sort of the job.
I'd agree that they're intimately linked and they complement each other. Like all of these things, it's important to validate those assumptions and understand those assumptions early on, which is something that in any case you would need to work closely with both the formulation and the device teams to understand where these assumptions come in. And I think that's something at TDP we do a lot. We enjoy that challenge of bringing those two together. And experimental can be quite limited at times, perhaps by, as you said, the work that's required to go into it, but also by some of the analytical methods. For example, we've seen formulations that are incredibly reflective, trying to do light-based particle counting or light-based analysis of homogeneity. Obviously incredibly challenging at that point because it looks like a mirror. Trying to build models in that can then undercut some of the limitations of an experimental setup can be incredibly powerful and incredibly enlightening. It's never a simple journey and it's never a sort of handover. I think traditionally it might have been seen that a formulation team has a proto-formulation and that proto-formulation has a certain level of it being fixed, and then it's for the device side to deliver that drug. I think certainly with the more challenging cases that we're seeing and with the want to try and get as much to market as we can, we know we lose a lot of drugs in the pipeline to tox results, to PK, to other clinical reasons. We really don't want to lose drugs in the pipeline for delivery reasons. We both have levers on the formulation side and on the device side. And to understand which levers complement each other and which levers should be pulled at what time, that's absolutely a collaborative process between both the formulation team and the device team. And certainly where we find it possible, we'd love to work with the formulation teams. We learn a lot. I think vice versa as well. It's an important relationship to keep going with.
I mean, in a given situation, there will be things that will be relatively speaking easier for them to fix, and things that will be relatively speaking easier for us to fix. And the only way of knowing which of those things are is by asking and doing it. And that is important, and that's quite enjoyable when you manage to make that work. I think it is challenging at times.
I mean, you can extend the metaphor that we usually use, certainly with something like a dry powder inhaler. It's a drug device marriage, and sometimes marriages are hard. But it is a combination product. That's what it's called. It's called that for a reason, because you are combining fundamentally a drug that needs to be delivered and a delivery method, and that delivery method needs to be tuned to the drug. And to do that, sometimes you have to make compromises on the drug side as well.
There is a great deal out there of sort of understanding of the way that these kind of particles behave in the literature and from experiments that can help us, I suppose. And it's sometimes a bit esoteric and sometimes a bit impenetrable, but it is there. Like it's not like it's impossible and doesn't exist and we have to invent it from whole cloth at this point. So I think that it gives me a fair amount of confidence that we can take that and we can use it and we can make it work, I suppose, with time and effort.
It's a great time to start building some of the toolkits for the future. We know at the moment there's a lot of formulation technologies that are in their infancy, and it goes to that driver to try and tune the release rate to get these long-acting formulations. And long-acting formulations in certain cases have been around for quite a while, but really there is a push towards it. A lot of these formulation technologies are in their infancy, but they're going to stick around. We want to understand how they behave. Some of those we understand relatively well now, but for some of those we don't, and there's a lot of learning to do. Building the analytical models that are underpinned by good quality experimental research are going to be very important in continuing to build the drug delivery tools that we not only use in the immediate future, but the long-term future as well. And so I share optimism in being able to deliver these. I think these are addressable challenges. But equally, I share optimism in building the groundwork for the future of this type of formulation delivery and where we can go with it. But also sharing an optimism in how we can improve things for the user. I think long-acting formulations is certainly a start. On the device side, we can do more. We can build in electrical mechanical features. We can start to apply our core understanding that's been developed and continues to develop over the years on user adherence, on usability overall, how people interact with drugs and with the devices as well, and start to build this holistic picture of how we can best serve these conditions with colloidal suspension.